Hello students, welcome to EPG Pathasala. I am Dr. P. Nagaraj, Assistant Professor from Anna University, Chennai. Today we are going to talk on model Protein Structure and its Functions from paper Structure and Function of Biomolecules. Objective of today's lecture is amino acids and its arrangements, protein folding and the type of proteins, structure of proteins, primary, secondary, tertiary and structure of proteins, binding site and its mechanism of proteins, antibody activities of enzymes, phosphorylation and its mechanism. These are all going to study today lecture. Of course, proteins make up of about 15% of the cells and have many functions in the cell, that is like enzymes, structural, transport, motor, storage, signaling, receptors, gene regulations, special function. These are all the functions of proteins. Shape, that is amino acid sequence, which is a primary structure of proteins. Proteins are made up of 20 amino acids linked by peptide bonds. Polypeptide backbone is the repeating sequence of NC, NC or CC in the peptide bond. The side chains are R group is not part of the backbone or the peptide bond. Polypeptide backbone, again polypeptide, which is nothing but peptide bond which is made up of combination of two amino acids will give peptide. More number of peptide will give combinedly polypeptide, also known as proteins. Suppose examples, glycine, alanine will give elimination of water to form glycalanine as a kind of peptide bond. Amino acids, there are we can majorly classify into two types, hydrophilic and hydrophobic. There are some amino acids which is belongs to hydrophilic, that means polar amino acids, and whereas certain class of amino acids are belongs to hydrophobic, which is also known as non-polar amino acids. Examples are aspartic acid, glutamic acid, arginine, lysine, histidine, asparagine, glutamine, serine, threonine, tyrosines belongs to hydrophilic or polar amino acids, whereas alanine, glycine, valine, leucines, isoleucines, prolines, phenylalanine, methionines, tryptophan, cysteines belongs to non-polar amino acids, also known as hydrophobic in nature. Protein foldings, the peptide bond allows for rotations around it and therefore the protein can fold and orient the R group in favorable position. Weak non-covalent interaction will hold the proteins in its functional shape. These are weak and will take many to hold the shape. So changing the shape, the position, that will vary the functional aspects. Non-covalent bonds in proteins, that means covalent bond which is made up of sharing of electron between two atoms. In non-covalent means it's a weak forces which may attract each other and make some kind of attraction or make a bond. In case of proteins, van der Waals force which is present, which is a very weak force, and also this polar group can make hydrogen bond. This kind of two very weak force of attraction present in protein molecules, which is called non-covalent bonds in proteins. Globular proteins, which is nothing but the side chains will help determine the conformations in an aqueous solutions. Proteins which is a globular, fiber protein, two types, which is a globular proteins. The side chains will fully determine the conformation of the particular proteins. Hydrogen bonds in proteins, of course, hydrogen bonds formed in proteins, either atoms involved in the peptide bond or peptide bond atoms and R groups or R groups. There are three types. Peptide bond involved in the peptide means that is a CONH, amide group. That amide group may be involved in hydrogen bonding, or peptide bond atoms at R group. That means that amide group either with the side chain or the side chain itself can make a hydrogen bond in proteins. Protein folding also shape is determined by the sequence of the amino acids. So some of them it may be helical, some of them may be the shape will vary. The final shape is called the conformations and has the lowest free energy possible, and then only that conformation will form. And denaturation is the process of unfolding the proteins. It can be down with heat pH, chemical compounds. In the chemical compounds can remove and have the protein renature or refold. This is an important phenomena. If the protein, which is three things specified, one is the temperature, substrate and pH. If you change any one, what will happen? Denaturation of protein will happen. If you add some chemical, also denaturation of protein will happen. Once if you remove the chemical, renaturations or refolding may happen. This is the specific criteria of protein folding. Refolding. Refolding is the phenomena. Molecular chaperones are small proteins that help guide the folding and can help keep the new proteins from associating with the wrong partner. That is refolding, which is opposite to folding. This purified proteins isolated from cells which is exposed to high concentration of urea, what will happen? Denature of protein occurs. The folding protein which is undergo refolding, that is denaturation. And if you remove the urea, it will come back again, the original conformations, that is protein folding. Refolding, folding, that may happen if addition of small chemical substance will make this refolding. Protein foldings to regular folding patterns have been identified in proteins that is formed between the bonds of the peptide backbones. Number one is alpha helix protein turns like a spiral, fibrous proteins. Examples of this kind of alpha helix fibrous proteins are hairs, nails, horns, etc. And another one is beta sheet. The protein folds back on itself as a ribbon or globular protein. That means the entire chain which is making like a ribbon like, and that one, that is make beta sheets. These two-way protein folding may occurs. Beta sheets again further explained by core of many protein is the beta sheets. It forms rigid structure with the hydrogen bond. Can be of two types. Antiparallel, that means run in an opposite direction of its neighbors, that is antiparallel. And another one is parallel, that means it's run in the same directions with longer looping section between them. This is how there are two types available in beta sheets. It form a rigid structure with hydrogen bond. That means it will not be flexible bonds. It cannot make only rigid bond. It can make because of this parallel antiparallel orientation. Alpha helix again, which is formed by a hydrogen bond between every fourth peptide bond of CONH, which will make the hydrogen bond. Every fourth unit making means it will make a spiral or helical bond, which is usually in protein that span a membrane. The alpha helix can either coil to the right or to the left, can also coil around each other. Coiled coil shape, a framework for structural proteins such as nails and skins. Levels of organizations, primary structure, secondary, tertiary and quaternary structures of proteins available. In case of primary structure, amino acid sequence of arrangement of amino acids in protein can be identified. In case of secondary structure, hydrogen bonding in the peptide chain backbone, further divided in two ways. One is alpha helix and another one is beta sheets. In case of tertiary structure, non-covalent interaction between the substituting groups within the protein. That is what the side chain present in the amino acids, that will have interaction like hydrophilic or hydrophobic interactions, which is also known as non-covalent interactions. Based on that, the orientation or flexibility of the molecule will change. That is what the tertiary structure of protein will give the informations. And of course, the last one is quaternary structure, the interaction between two polypeptide chains. That will give you quaternary structure of the proteins. Protein structure which involves a simple as a straight chain, which is a primary structure, and the second one is secondary structure, which again alpha helix and beta sheets, and the third one is a single polypeptide domain, which is looks 3D, and the fourth one is proteins molecules made up of two different domains, means two protein molecules all interact together, which will give the quaternary structure of the proteins. A protein having active sites that is also known as domains. A domain is a basic structural unit of protein structure, distinct from those that make up the conformations. Part of proteins that can fold into stable structure independently. Different domains can impart different function to proteins. Proteins can have one to many domains depending on protein size. So the domain is very important, that is active site, whatever biochemical reaction that may happen in the domain. So based on the size of the proteins, domain size or numbers will vary depend upon the size of the proteins, and the function also vary accordingly. The domains which is again gives more information. The domain which is not present in case of primary structure, whereas a secondary which is least preferred in domains, whereas in tertiary and quaternary structure the domain play major role, and that domain which is involved for many biochemical reactions or interactions. Some of the useful proteins are thousands and thousands of different combination of amino acids that can make up proteins, and that would increases if each one had multiple shape. Proteins usually have only one useful conformations, because otherwise it would not be efficient to use the energy available to the system. Natural selections has eliminated proteins that do not perform a specific function in the cell. So this is very important, that conformation, it's a easily available conformation without giving any external energy. That conformation act as a domain. All the biochemical reaction may occur in the particular domain or conformation aspects. Protein families, there are two types, elastase and chymotrypsins, have a similarities in amino acid sequence and 3D structures, have a similar function such as breakdown proteins, but do it differently. This is how the protein families or protein-like compounds can do actions. Proteins, multiple peptides. Non-covalent bonds can form interaction between individual polypeptide chains. Binding site where the protein interactions with one another. Subunit each polypeptide chains of large proteins. Dimer proteins made up of two subunits. This can be same subunit or different subunits depend upon based on the interactions. Single subunit proteins, which is shown here, will interact each other, will make a tertiary structure. If more number of subunit interact each other, will give quaternary structure of proteins. Different subunit of proteins, suppose here two type of beta globulins and two types of alpha-globulins interact each other, will make a complex molecule which is hemoglobin. That there are 4 subunits of protein which is combined together will give hemoglobin. Out of 4, 2 alpha globulin subunits and the other 2 are beta globulin subunits, which will give complete proteins. Protein assemblies. Protein can aggregate each other to make a assemble together, will make a chain or circle like a structure. Proteins can form very large assemblies. Aggregation happened, can form long chains if the protein has 2 binding sites linked together as a helix or a ring. Actin fibers is the muscles that cytoskeletons is made from thousands of actin molecules as a helical fiber. This is how proteins can aggregate or assembles together will form a helical structure. Types of proteins are, we know, 2 types major, globular proteins and fibrous proteins. The globular proteins are most of what we have deal with so far is compact shape like a ball with irregular surface. Enzymes are globular. In case of fibrous proteins, usually span a long distance in the cell. 3D structure is usually long and rod shaped. These are all types of proteins. Important fibrous proteins, intermediate filament of the cytoskeleton, structural scaffold inside the cells, example keratin in hair, horns in nails, etc. Extracellular matrix, bind cells together to make tissues, secreted from cells and assembled in long fibers. Collagen fiber with the glycine, every third amino acids in the proteins. elastin, unstructured fibers that gives tissue and elastic characteristics. Collagens and elastins can be explained. Collagen is the molecules, the size short section of the collagens fibers. Collagen's triple helix can form together, whereas in case of elastic fiber, which can be expanded. Stabilizing cross-linking. Cross-linking between proteins which is made up of sulfur present in the amino acids. Two thiol unit combined together, elimination of hydrogen will give sulfur-sulfur bond, which will give the cross-linking. The cross-linkages can between the two parts of the protein or between two subunits. Disulfide bond, also known as SS bond, formed between adjacent SH groups and the amino acid cysteines will make or stabilizing the cross-linking. Proteins at work. The conformation of protein gives its unique functions. To work, protein must interact with other molecules, usually one or few molecules from the thousands to one proteins. Ligand, the molecule that a protein can be bind. Binding site, part of the protein that interact with the ligand. It consists of a cavity formed by a specific arrangement of amino acids. That specific cavity can interact with either one or two other molecules. Given the structure of ligand binding, where the specific cavity available in the protein molecule, where incoming ligand can come and fit into the protein molecule to make non-covalent bonds, will give close together. Formation of binding site. Binding site nothing but the cavity. The binding site forms when amino acids from within the protein come together in the folding. Folding or changing the structure will give the binding site. The remaining sequence may play a role in regulating the protein activities. That is what the amino acids can arrange themselves to make a cavity based on the substrate. After that, the rest of the part will take the regulating the protein's activities. So antibody family. What is antibody? A family of proteins that can be created to bind to almost any molecule. That kind of molecule which is called antibody. And antibody immunoglobulins are made in response to foreign molecules like a bacteria, virus, pollen, etc. are called antigens. Bind together tightly. That antibody, that means the enzymes which will bind together very tightly with either bacteria, virus or pollen, and therefore the inactivate their antigens or marks for their destruction. That is, it is killing. That effect is because of the shape. Normally antibodies which is having Y-shaped molecules with the two binding sites at the upper end of the Y. So the loops of the polypeptides on the end of the binding site what imparts the recognition of the antigens. Changes in the sequence of the loops makes the antibody recognize different antigen specificity and which is inactivate that particular antigens. Antibodies, again, which is Y-shaped antibodies. Antibodies as the protein form, it will make an active site where antigens, bacteria, virus, it will come to the antibody and it will mix together and it will inactive the activities of this virus or bacteria. Binding strength. The binding strength can be measured directly. Antibodies and antigens are mixing around in a solution. Eventually, they will bump into each other in a way that the antigen stick to the antibody. Eventually, they will separate due to the motion in the molecule. The process continues until the equilibrium is reached. Number sticking is constant and the number leaving is constant. This can be determined by any protein and its ligand. So this is a kind of antigen-antibody interactions. The protein molecule acts as antibodies, whereas the virus or bacteria will be antigen. It will come and interact. The activity of virus and bacteria can be inactivated due to presence of antibodies. Equilibrium constant. The concentration of antigen antibody and the antigen antibody complex at equilibrium can be measured. Equilibrium constant. Larger the K equilibrium constant, the tighter the binding or the more non-covalent bond that hold the together. Enzymes as catalyst. Enzymes are proteins that bind to their ligand as the first step in the process. An enzyme ligand is called substrate, maybe one or more molecules. Output of the reaction is called the product. Enzymes can repeat these steps many times and rapidly called catalyst. Many different kinds available. Enzymes at work. Lysozyme is an important enzyme that protects us from bacteria by making holes in the bacteria cell wall and causing it to break. So this is a very important useful work of enzymes. Lysozyme adds water to the glycosidic bond in the cell wall. Lysozyme holds the polysaccharides in a position that allows the water to break and bond. This is a transition state, a state between substrate and product, that is called a transition state. Active site is a special binding site in enzyme where the chemical reaction takes place. That is what active site or domain where the chemical reaction, really biochemical reaction takes place, that particular site. Lysozymes, which is again non-covalent bonds, hold the polysaccharides in the active sites until the reaction occurs. So which is non-covalently, it will make a cavity, then whatever the substrate, it will substrate plus enzyme will produce enzyme substrate complex, which is further produce enzymes product complex, then finally enzyme is separated from the products. Features of enzyme catalyst. Enzyme binds to two substrate molecules and orients them precisely to encourage the reaction to occur between them. It will orient or reorient themselves to adapt the, depend upon the antibody, it can make the reactions. Enzyme performance. Enzyme plus substrate will give enzyme substrate complex, which will further give enzyme product complex, which will further give enzyme will be evolved plus product also given. Step 1, binding of the substrates, limiting the step operating on substrate and enzymes. Vmax, maximum rate of the reactions. Turnover number determines how fast the substrates can be processed. Rate of reaction E. Step 2, stabilizes the transition state. State of substrates prior to the becoming product. Enzymes lowest the energy of transition state and therefore acceleration of the reactions takes place. Reaction rate. Reaction rate that allows reaction to proceed at half of its maximum rate. Prosthetic groups. Occasionally the sequence of the protein is not enough for the function of the proteins. Some proteins require a non-proteinous molecule to enhance the performance of the proteins. Hemoglobin requires heme, ion containing compound, to carry this O2 or oxygen. When a prosthetic group is required by an enzyme, is called coenzymes, usually a metal or vitamins. These groups may be covalently or non-covalently linked to the proteins. Regulation of enzymes. Regulation of enzymatic pathway prevent the deletion of the substrates. Regulation happens at the level of the enzymes in the pathway. Feedback inhibition is when the end product regulates the enzymes early in the pathway. Allostery, the conformational coupling of two widely separated binding site must be responsible for regulation. Active site recognize substrate and second site recognize the regulatory molecules. So these two are very important to do the work functions. Protein regulated this way undergoes allosteric transitions or conformational changes. Proteins regulated in this manner is an allosteric proteins. Another class of important reaction is phosphorylations. Some proteins are regulated by the addition of a phosphate group that allows for a attraction of positive charges side chains, causing conformational change. If you take one protein, just add some phosphate, the conformational changes will be there because of attraction of that positive charge. Reversible proteins, phosphorylations regulate many eukaryotic cell function during this on or off. A protein kinase add the phosphate and protein phosphate remove them. So this is how the phosphorylations takes place. Once the conformation changes, then the activity also will change. GTP binding proteins. also known as GTPases. GTP doesn't release its phosphate group, but rather the guanine part binds tightly to the protein and the protein is inactive. So hydrolysis of the GTP to GDP, the protein itself, and now the protein is inactive. Also, a family of proteins usually involved in cell signaling, switching proteins on or off. This is how the binding proteins can function itself. The binding site which is inactivated by using these proteins. Proteins machines, complex of 10 or more proteins that work together, such as DNA replications, RNA or protein synthesis, transmembrane signaling, etc. Usually driven by ATP or GTP hydrolysis. So students, let us now summarize what we learned in this module. Amino acids and its arrangement, sequence of arrangements, and protein folding and the types of proteins, structure of protein, of course, primary, secondary, tertiary structures of proteins, we learn details. Binding site and domain are active sites and its mechanism of proteins that we studied in details. Apart from that, antibody antigen activities of enzymes that we studied in more elaborately. Phosphorylations is the important phenomena of changes the protein conformations and its mechanism also we studied in details.